So we're going to talk about population ecology and survivorship curves today. I'm going to talk about the different types of survivorship curves and what they mean. And in order to understand the survivorship curve chart, we're going to have to learn a little bit of exponential growth and semi-log, how to use semi-log paper. But using semi-log paper, learning how to use it and learning how to write exponential growth and what that means has never been more useful than in the time of COVID-19 in which we find ourselves in. So you're going to learn some stuff you never knew you wanted to learn, and you're going to be able to interpret some curves that you probably interpreted slightly differently until this class came around. So happy learning today. First things first, we're going to have to talk about a couple of definitions. First, population. A population is a group of individuals at the same place, of the same species, found at a particular place. And each species has a unique pattern of development, a unique reproductive rate, migration, and survivorship. Now, we measure survivorship as a way of counting mortality by looking at the individuals that survive. So we're not looking at the dead ones. Because sometimes it's hard to find the dead ones, they get eaten, they disappear. So survivorship is a way of looking at the ones that live. And the experiments we're going to do, we're going to be doing those by looking at the creatures. Well, they're not going to be creatures for us, but the things that survive. When you're looking at different species, they have very distinct different types of survivorship curves. There's one type of survivorship curves that's type 1. Humans are an example of that. In this case, you have a high survival in the young, so babies tend to survive, and these creatures tend to die in old age. If you ever try to remember which one is type 1, type 2, and type 3, just remember type 1, that's us. That's because we're number 1. Survivorship type 2, there's an equal survival of living or dying pretty much over your lifetime. Your example of that is corals, birds, mice, that kind of stuff. And we'll talk a little bit about what causes you to be a type 2. Type 3 is where the death rate of the young is very, very high. And as you get older, your odds of dying actually go down. So the examples of that are plants in general, insects, a lot of insects anyway, and fish. Survivorship curves are not something that we came up with. They're just plots that represent what we observe. And these plots of what we observe represent different strategies for survival. There are different ways of being successful for your species to survive. And each one of the surviving species we have on this earth today managed to be successful in some way, shape, or form. And survivorship, or the survivorship curve that works for them, has something to do with how they reproduce and how they develop and what they do to be successful. So let's look at some of these pictures and let's see if we can figure out what kind of survivorship curve this creature probably has. So on the top right, you see a whole bunch of little froggies. Um, what kind of survivorship curve do you think that that may be? Would that be a 1, a 2, or a 3? And after you think about it for a moment, you should come to the conclusion, hopefully, that if you have a whole bunch of youngsters, that probably means that a lot of young ones of your species die. Um, so that's the case here. So if you have a lot of um, young ones die of your species, you have to create a lot of them. So in the case of frogs, they tend to lay eggs, and then when all the little what do you call them? The little tadpoles come out of the eggs. Everybody and their brother eats them. And so very few little frogs actually make it to a larger frog. Similarly, what you've got in the middle, that whole clutch of eggs there, massive amounts of eggs, what organism is this? Can you tell? And what those are turtles. Turtles is another example of a type 3 survivorship curve. They lay a bunch of eggs. Mom buries them. She doesn't really take care of them or anything. She just walks away. And, the little turtles are, good luck with life. And then top left, you see where the little turtles came out of the shell and are trying to make their way to the ocean, where they're probably greeted by predators that would like to eat them like right now and immediately. Now, looking at the bottom right, what's different about that picture compared to both the way the method that the turtle uses or the frog? And what's different is pretty obvious. You've got a mom there who takes care of her little pup. And because of that, because of the caretaking, the young tend to survive. There's a caveat though, right? Because if your young need to survive and you need to take care of them, you can't take care of 1,000 of them. You can take care of maybe 10 or so on a good day. And the other example we have on the bottom left is a bear. A grizzly bear also is a type 1 survivorship curve. They have very few young, but they take care of them. And because they take care of them, it's very likely that the bear cubs will survive, which is very similar to the way we do this with humans, right? Now, when you look at the squirrel in the middle there, what type of survivorship curve do you think a squirrel may have? Squirrels do take care of their young. There's a nest of young that they have, and they do, in fact, take care of their little guys. However, when you looked at the previous slide, mice also take care of their young. And mice were still considered to be survivorship curve 2. And I would say the same thing is true for squirrels, because squirrels have a dangerous life. They can fall out of the nest. Have you seen the high wire acts they do when they climb across all the telephone wires? Everybody and their brother wants to eat squirrels, and little squirrels die a lot. So chances are for a squirrel, crossing the street and all that, chances are for a squirrel, that's also a survivorship, type 2 survivorship. And if I ask you in an exam which survivorship curve something has, a species has, I will give you a very clear example. It's going to be either, a turtle or a puppy. I will not likely ask you to do a squirrel. And if I do, I will allow you to argue your point. And if you can argue your point, well, then, I'll give it to you. Which brings us to the next organism, bees. Now, on the previous slide, it said that insects generally are type 3 because you have a lot of them, and then, you know, they're very likely to die. Bees, I would allow you to argue both a 2 and a 3. Because while there are a lot of them and they make a lot of babies, bees also take care of each other as much as they can. They will clean each other, they will take care of each other, they will defend each other. And so, because of the hive structure, I would say that a bee, you could argue both a 3 and a 2. The thing you may not have paid attention to when you looked at the survivorship curve was that the y-axis actually was labeled with the word log number of survivors. And the log number of survivors means that what you were graphing there was actually exponential growth. So what we're going to have to talk about now is exponential growth. And to tell you about exponential growth, I'm going to have to tell you a story. So there's the legend of the guy who invented the chessboard and the king. And this legend is told both with rice grains and with wheat grains. There is no difference with the legends whether you're using rice or wheat. So what this picture shows is basically the beginnings of the story. So when chess was invented, the king was really impressed with that game, and he told the guy who invented the game, "Hey, I'm going to give you anything you want." And the guy who invented the game said, "Great. So what I want from you is I want you to give me 1 grain of rice for the first board of my chessboard, 2 grains of rice for the second, and then double it, 4 grains of rice for the third, double it again, 8, 16, 32, 64, and for the last field of the first row, it's going to be 128 grains of rice. And I want you to double the rice until we get to the 64th field. That's what I would like." And the king goes, "Oh, that's easy." And so on day 1, he gives the inventor of the chessboard 1. On day 2, he gives him 2. On day 3, he gives him 4, and so on and so on. How much rice will he give this person at the end of the game? So at field number 64. I'm going to give you a minute to try to think about that before I'll switch to the next slide. So you put 1 grain of rice on the first square. The next time you double that number on the second square. That's 2 grains. The next day you double that number on the third. That's 4. Then you double that number on the fourth. That's 8. How many grains are on the last square, which is square number 64? On the 64th square, you would have 2 to the 63rd number of grains. And that is that huge number right there that starts with a 9 that I'm not even going to start trying to name. And the whole board would contain almost twice that. That's approximately 461 billion metric tons of rice, which would be a heap of rice that's as high as the Mount Everest. The annual production of rice in India in 1992 was only about 100 million tons. So it's a lot more than that. So needless to say, the way the story goes is they ran out of rice before they could actually pay off the inventor of the chessboard. So when we talk about exponential growth, we need to talk a little bit about exponential growth in business because that's how pyramid schemes work. And that's why pyramid schemes don't actually work because you're literally running out of people. By the time you're at the 11th generation, if you're going with a multiplier of 6, and the article explains this to you that's linked underneath of that picture, you're running out of the population in the US. And by the time you get to the 13th level, you're running out of the number of people in the world. However, exponential growth works in nature. Bacteria show exponential growth, which is why scientists learned how to graph exponential growth. As long as bacteria get enough nutrition under ideal condition, they can produce more and more and more bacteria. So if you give them enough nutrients, they will keep growing and they will keep doubling every 30 minutes. And when you have bacteria on a plate, you remember watching bacteria under the microscope, you could barely see them. And you can go from one bacterium, if you put one bacterium on a plate, that on the right-hand side is a bacterial plate, it's a petri dish. And what you do there is you streak out bacteria trying to get isolated colonies. And so you literally, those little dots that you see there, those macroscopic colonies, those represent about 2 to the 24, 2 to the power of 24 bacteria. So about 10 million bacteria that are actually visible with the naked eye. And you can pick them up and streak them out again. So you've gone from something that you can barely see under the microscope to a visible colony in one day because the growth, the exponential growth, is so powerful and so fast. So back to the rice and the chessboard problem. When we're going to try to graph this, we're going to have an issue. And this is all the same graph. It's all the same numbers. They're just graphed in a different way. I'm trying to get all the numbers onto a graph. So the first one on the left, you only get into generation 6 to day 6, right? You're getting 2, 4, 8, and you're getting up to 32. But that really doesn't show you the massive amount of growth you get, right? On the second graph, you're going to to day 11, but you're completely losing, or you're starting to lose, all the numbers from day 1 to 5. You really can't distinguish the numbers anymore because by the time you get up to 1,000, your 1 through 10 is gone. And that problem becomes bigger the greater your numbers get, the higher your numbers get. And basically on the last graph, you may be able to tell where your numbers are at on day 29, but you have no prayer at trying to discern what the numbers look like from day 1 through day 25, really. So how do we solve this problem? How do we solve the problem of trying to graph this in some way so that it's representative, but it also gives you an idea of what the numbers are across the whole length of the, across all the days. And the way this is done is the reason why some scientists get paid the big bucks. So these two curves, the one on the left and the one on the right, are actually the same numbers graphed. What's changed, the x-axis is the same, but your y-axis changed. Your y-axis went from a linear scale to a log scale. So you're still describing the number of grains, but on the left-hand side, you've got a linear scale. On the right-hand side, you have a log scale, which means on the right-hand side, you're going from 1 to 10 to 100. So each line is a factor of 10. And by doing that, you can then, you get, the magic is you get a straight line. Scientists love straight lines because with straight lines you can extrapolate, right? With a straight line, you can use a ruler and you can then say, if I ask you, what is this on day, I don't know, 64? I can make a line that goes all the way to day 64 and I can tell you what that number is because it's just, multiply by 10. And then, if I can figure out how to verbalize this number, which I don't, I can tell you what that number is. Where I encountered exponential growth for the first time in my life was when I was looking at bacterial growth. And you've probably, if you ever talked about bacteria in class, you talked about the log phase and the stationary phase of the bacterial growth. And what that is the same thing we've just discussed. Where bacteria double under ideal conditions, that's called, it's in the log phase, which means it creates a straight line on semi-log paper. You recognize semi-log paper because semi-log paper has this funny way of looking. It's got these cycles where the lines start out kind of wide, closer to the x-axis, and then they get more and more narrow, and then they go up again. And it's these cycles where you go from a wider distance between your lines to a very narrow distance between your lines. And each set of these is a cycle. Now the label on the left-hand side of this semi-log paper is not a very good label because it should actually be, the first one should be a 1, then the next one should be a 10, and then 100, and then 1,000, because each one of these always represents a factor of 10. But we'll get to that in more detail in just a second. But at any rate, the log phase is where the bacteria double, and then the stationary phase is where the bacteria run out of nutrients, and the growth slows down for some reason. But anytime you have semi-log paper, and it's semi-log because the y-axis is in a log scale and the x-axis is in a linear scale. That's why it's called semi-log, because it's half log and half linear. Now, exponential growth, they come in different flavors, right? Exponential growth can be 2 to the n, or 3 to the n, or 1.5 to the n. And all of those exponential growth curves make a straight line on semi-log paper. What's different is the steepness of the curve. Now this semi-log paper you see here actually has the cycles labeled correctly. So you start out with 1 on the first cycle, you get to 2, 3, 4, 7, and 10. Then you get to 10, and that next 2, well, okay, it's sort of labeled correctly. That next, the 2 there, that's not a 2, that's a 20. And then you have a 30, a 40, 70, and 100, and then it's 200, 300, 400, 700, and 1,000. The depending on what the base number is of your exponential growth, it depends on the steepness of the curve. I don't know if I said that already, but yeah. So for your homework assignment, you're going to label semi-log paper, and you're going to have to use semi-log paper. So the first thing I want you to do is to figure out how many cycles are on the sheet of semi-log paper, because they come in different flavors. They come in 1 cycles, 2 cycles, 3 cycles, and I think I've seen up to like 8 cycles on one sheet of semi-log paper. So when you're looking at semi-log paper too long, your eyes go cross. And usually I find that it's better if I just hold the piece of paper far, far away from me. And the first thing I do is just decide how many cycles there are. And on this particular paper, you have 3 cycles. Now, it's 3 cycles because there's 3 things that are, it's triple what you're seeing. Now, when you're looking at the very bottom, it's very light, then it's getting darker where the lines are getting more narrow and narrow, closer and closer together. And then it lightens up a little bit. That's an illusion because what's happening here on this piece of paper is in the first 4 cycles, oh, not 4 cycles, in the first 4 subdivisions of the first cycle, you have a line for every number. But then there's not enough room for a line for every number, so then you have a line for every other number. And that's why each cycle looks like it's got a darker and a lighter shade to it. But if you're looking at the overall paper, which is why I'm suggesting that you hold it far, far away from you so you can see what the actual repeat is. So this is 3 cycles, and the piece of paper that you've got in your handout, this is actually a copy of the piece of paper that you have in your handout. So the piece of paper you have in your handout is a 3-cycle semi-log paper. After you decide how many cycles you have, the next thing you got to figure out is how to label them. And my advice would be to keep in mind that each cycle is a multiple of 10. So on the right-hand side here, you have a different type of semi-log paper with, again, 3 cycles. And each cycle can be a multiple of 10 of, is a multiple of 10 of the other, but there's no rule that you have to start with 1 at the bottom. So you could, for example, start on the bottom with 10 and then go to 100, 1,000, and 10,000. Or you could go 1, 10, 100, 1,000. Or, 1,000, 10,000, 100,000, 1,000,000. All of that is possible. So that would be when I'm asking you to label semi-log paper, do that first. Go and decide what your range is, what numbers you're trying to cover. In our case in the lab, we're gonna try to label percentages. So your highest number is going to be 100, which means you have to have at least a cycle for 1, a cycle for 10, and a cycle for, well, that goes up to 100, which means you really only need 2 cycles, but you have 3, so you can make a choice whether you start at the 1 or you start at 0.1 for the first one. But then in between, you're labeling, so from the 1, you're labeling 2, 3, 4, all the way to 10, and then the next line after the 10, that would be a 20. Because you're in the next cycle goes from 10 to 100. And we're going to practice this a little bit during class today. And you may be asked in the exam to actually label one of those yourself. Now, as you're sitting here, most of you are pretty sure you've never seen one of those semi-log graphs before in your life. And you'd be wrong. Because if you've seen Andy Bashir recently, this is actually the graph he shows where he talks about cutting the top off the muffin. This is new cases, weekly average with Kentucky, and this in fact is semi-log paper. You recognize the cycles? And when you look on the left, you see the 1 and the 10 and the 100, and then you've got the 1,000, right? But we didn't get that far, so But yeah, so we are looking at semi-log paper all the time these days. And I want you to look at a couple of those now, just because I think it's interesting to see how you can make data look different just by the way you present it. And you've got to be very careful when you're looking at how people represent data, um, because if they want to make a point, they can represent data in one way or another, and the same data looks very, very different. So let's look at a couple of graphs that are actually identical. They're just presented different. On this slide, you have two graphs that are actually different, but they're just represented differently. So the top two are cases by country. So you've got the United States highlighted, and the rest of it is the top 10 countries. And the bottom, you have two graphs that are showing cases by country, but normalized by country population. And in each case, the one on the left is the linear one, and the one on the right is the log one. So when you look at the top ones first, the cases by country, the one on the left, it looks, when you're looking at the, so look at the trend line, because the trend line is the same for all of them. So when you look at the trend line on the one on the left, it looks almost like United States has like almost no cases at all. And then it very quickly leaves the trend line, but it looks like the United States has like way more cases than anybody else. And it looks, it looks better than the trend line, but it looks pretty bad compared to everybody else. When you look at the one on the right, and this is the same data, this is absolutely 100% the same data, you realize that what you saw on the left, where that first part, where it looks like there was really no cases at all until about day 20, that's actually where the exponential growth was happening, right? Because that's where you have the almost straight line on the semi-log paper. And it's not until about almost day 25 where the curve in America flattens off away from the exponential growth, which is probably when we started using social distancing. The one on the right also, there's two take-home messages from the one on the right. The one take-home message is that on the semi-log paper, the distances between, like the distances between the lines are compressed. So your distance between Italy and the United States is not very much, even though the numbers are a lot higher. And that's because when you get higher up in the semi-log paper, the distance between 10,000 and 100,000 higher up is the same as the distance between 1 and 100, because of the way semi-log paper is set up. So that's kind of the look at the ones on the top, and I gave you the page where you can play with all the data, just so if you wanted to look at what different things look like, and what you can highlight and all of that. Now the one on the bottom is a different set of data. It's again the top 10 countries. However, it's the cases by country normalized for the population. And when you're normalizing it for the population and you're just looking at the top 10 countries, if you look specifically at what the countries are looking like, the one on the left, the linear one, the United States actually looks pretty good, right? It's tracking right along with Italy, and it's one of the lower ones of the cases. But if you look at who the countries are that do badly on that graph on the bottom, it's the really small states. that have real problems, the little city states, because just a few cases in a little city state um, has a huge effect. Like, so if you've got your Luxembourg or your I think it's Marino or Monaco or whatever, is that's part of that list. So it's really not a very good comparison. So you got to be very careful what numbers you're looking at. Um, the one on the right again shows, just like on the top, it shows that the United States, even with looking at the numbers normalized for population, that we initially had exponential growth. Now it's not an exponential growth that's a doubling, but it's whatever the trend line is. And in both cases, we're tracking right along with Italy, which we know Italy had a disaster at their hands. So, but yeah, so I just wanted you to look at these numbers, and if you so desire, you can go to the website and just play with numbers and play with graphs and see what these different things look like based on how you're deciding to represent them. And the take-home message from this sheet is, number one, is you always have to look at the axis. You always have to look at the axis on the left, because it tells you a lot about how the graph is set up. And it makes a very different impression in terms of a graph, whether you have a linear or you have a normal, not normalized, a logarithmic curve. And then obviously being normalized for population is super important for something like the United States. Because if we have more people than everybody else, obviously we're going to have more cases. Of course, the other thing you got to keep in mind for us is we have a shortage of tests. So whatever numbers we have are going to be handicapped by the number of tests. And all of this data, you can lie with data. Unless you know exactly where your data is coming from, you never know if you're comparing apples and oranges, especially in a situation like right now, where we're dealing with data where every state has different access, every state has different rules, every country has different access, different rules, different ways of testing. So really, it's hard to compare anything to anything. But semi-log paper will give you the ability to see something over the entire curve. So if you have very big numbers, semi-log paper will allow you to look at a curve from the very bottom to the very top and see the shape of it. And if it's a straight line, then it's some kind of a logarithmic growth. Whereas with a linear curve, as you see on the left, you're losing either the bottom end or the top end, because one of them can't be really discerned, because there's no way to represent, to make this thing tall enough that you can see everything. And last but not least, let's look at this paper here real quick. So can you tell me how many cycles you have here? And if you can tell me how many cycles you have here, if I tell you the A is a 1, what numbers are the B, C, D, E, and F? Can you tell me? And what if I tell you the C is a 1? What are the other numbers? Can you tell me? And what if I tell you the A is a 10? Can you tell me what the other numbers are? And you should be able to figure this out for every one of those, which numbers they are. Because chances are you're going to see something like this on the exam, and I'm going to ask you to figure it out.